So this is just a review of renal physiology. If you feel really comfortable with renal physiology, you can skip this. And I'm actually borrowing a lot of the concepts that I'm going to kind of go over here, and especially how it's presented from the Khan Academy. That has a great overview on the urinary system, and if you want to skip straight to the source, which is the Khan Academy videos, I highly recommend those, and that's KHAN. So I'm going to start with the glomerulus. We've talked some about the glomerulus, realizing that the afferent arteriole is what kind of comes into the kidney, right? And then it swirls around and then comes out as the efferent arteriole. So that's efferent and afferent. And of course this is fed by the renal artery. And this is what allows for glomerular filtration. So the greater amount coming into the afferent arteriole, the greater pressure there is, the more that is going to cross over that space into what's called Bowman's space. And that's kind of this collecting area after the glomerulus. So taking a small snapshot, right, of, if you imagine this area right here, and just one vessel in there, and then this is where we're going to look at it. So that's the vessel, this area right here, and then this is kind of this Bowman's capsule or Bowman's space. So that's what we're going to look at to talk about glomerular filtration. So the blood, which of course this is our capillary, our arteriole, the greater the amount of blood and the greater the pressure, it pushes fluid across the glomerulus. And so what the glomerulus is this vessel has blood, right? So there's endothelial cells and they're fenestrated, meaning there's holes kind of in between the endothelial cells. And that's what actually allows, that probably shouldn't be blue since it's blood, that's what allows the blood to kind of cross over. So that's one layer. There's also a basement membrane, which will make a different color. So there's a basement membrane in here, and then there are cells from the renal tubular cells, and they have these projections called podocytes. And so this three kind of membrane thing is what makes up the glomerulus. And it is essentially a semi-permeable membrane, which allows for cells to stay within the vessel. So our cells actually stay within the vessel. Our red cells stay within the vessel, and our white cells stay within the vessel. Sorry, they're the same size. And really large proteins stay within the vessel. And large proteins are albumin. Now, of course, the fenestrations would allow them to go through, but there are special things within these membranes, within, the basement membrane and the podocytes, which actually keep the proteins from going through. And so what actually gets through is what we've talked about before, which is a cell and big protein-free filtrate of the plasma. So what does that mean that it actually is containing? Well, it means it contains electrolytes, so things like sodium and chloride and potassium, bicarbonate, ions such as hydrogen ion. It contains waste products such as urea and creatinine, phosphorus. It contains things like glucose and amino acids. And it also contains very small proteins, so these guys are very small. And of course, why it's important that you realize what's actually in this filtrate is because normally most of these substances are reabsorbed. Some of them are waste products, such as creatinine, some of the urea, some of the phosphorus, a lot of the hydrogen ion. But most everything else needs to be reabsorbed. And the reason it has to be reabsorbed is because it's all much smaller than urea and creatinine, for example. So everything else, since it's in a smaller amount, gets through with urea and creatinine and then has to be reabsorbed later with, of course, a lot of water, because there's a lot of water that's actually going and being made into the filtrate. And so that's where, when we talk about the tubules, which is the next part, so the functional unit of the nephron, again, is the glomerulus, and then plus the tubule. So that's our nephron. So let's move on to the tubular part, and we're going to start with the proximal convoluted tubule. So the proximal convoluted tubule is very metabolically active, and it reabsorbs a tremendous amount of products. And these include the majority of things that actually just got through from our filtrate. And this includes water, sodium, chloride, bicarbonate, things like glucose, amino acids, and numerous other substances, which I'm not going to go through all of them. I'm just going to try and focus on the ones that are going to matter when we talk about renal tubular damage. And that's because the proximal convoluted tubule, which is very convoluted, is very metabolically active, so it's very likely to be affected by damage. And this, again, reabsorbs about 65% of the solutes and water that was presented from the filtrate. So this next part, of course, is our loop of Henle. And in the middle of our loop of Henle, approximately in the middle, you can kind of separate things out into the cortex and the medulla of the kidney. The cortex being the outer part, the medulla or the middle part. And our loop of Henle has a descending limb, which obviously go this way, and an ascending limb, which goes this way. And we call this whole process countercurrent multiplication. I'm going to tell you what that is right now. The medulla is also, we consider, well, not necessarily just the medulla, but we consider kind of this area outside of the tubule, the interstitium. And so when we talk about mostly the medullary interstitium, I'll write it over here for ease. So the medullary interstitium is what's actually surrounding our tubules. So that's our medullary interstitium, and that's going to become important in a second. So within our ascending loop of Henle, we actively reabsorb sodium and chloride. And that makes our medullary interstitium very salty. So we're going to put lots of little salty things in here. And we make it very salty so that in our descending loop of Henle, we can passively reabsorb water. So this is a passive process where we reabsorb water to facilitate water reabsorption in our actual kidneys. So again, the ascending limb is what is allowing us to actively reabsorb sodium and chloride, and our descending limb is what is allowing us to passively reabsorb water. And of course this is called countercurrent multiplication because it's this, again, active reabsorption of sodium and chloride that allows for the reabsorption of water passively because of how salty this kind of background medullary interstitium is. Okay, so now we're going to move on to the distal convoluted tubule. So I just moved the efferent arteriole to be a little bit more lifelike. Okay, so our distal convoluted tubule actually goes right by our glomerulus, and so that area right there, so this area right here, is your juxtaglomerular cells, and this is what impacts, again, the body sensing blood volume and determining blood pressure, and what relates to the renin and angiotensin system, and we're not going to go into that a lot. And so the distal convoluted tubule is very important in reabsorption of water. And prior to this distal convoluted tubule, essentially all of the tubular reabsorption of water had occurred without regard for the body's needs. It's essentially automatic. But once we get to the distal tubule and the collecting ducts, now it's going to be very, very dependent on aldosterone and ADH. And so only 9% of the original filtrate is remaining in the tubules once it gets to this early distal tubule. So it still means that you could become rapidly dehydrated. It's just that this is the part that's going to be dependent on what's going on in the body. And in this distal convoluted tubule, now aldosterone is going to take effect, if need be. And aldosterone means that sodium and chloride will be reabsorbed along with water in the face of aldosterone, and potassium is going to be excreted. And this means anytime's reabsorbed, it's being reabsorbed outside of the tubules. And if it's being excreted, it's put into the tubules to be made as urine. Now, many distal convoluted tubules eventually join up, and they enter into what's called the collecting duct. And so these are little areas where there are other distal convoluted tubules that are feeding in. And within the collecting duct, ADH exerts its effects. And of course, ADH can reabsorb water, and it can do it in the face of urea. So urea will be reabsorbed along with water, or just water is going to be reabsorbed. So urea is not always reabsorbed, but water is reabsorbed. So ADH is reabsorbing water. So another thing that happens with the efferent arteriole is that it branches off into small capillaries, and these sort of traverse the various parts of the renal tubule. And these are called the peritubular capillaries. And their job is to actually reabsorb all of the good things that the kidneys have actively reabsorbed from the filtrate. And so this will eventually rejoin and then go to the renal vein and enter back into circulation. And that is why you then don't lose any of those things that your tubules reabsorb. Anything that your tubules don't reabsorb, of course, becomes a waste product and goes out then through your kidney into your renal pelvis, out through your ureters and into your bladder so that you can pee it away. So a few comments just on the difference between glomerular filtration and the tubular function. So the glomerular filtration is determined by how much blood actually is getting across each glomerulus. And remember that we're talking hundreds of thousands of nephrons within the kidneys, not just one. And so glomerular filtration is related to some pre-renal factors. So what is your actual volume of blood? So your hydration status, your actual blood volume, if you're hypovolemic, what your cardiac output is, what your blood pressure is. Those are the primary ones that determine kind of those pre-renal factors, so that if you have a decrease in any of these, that's going to lead to a decrease in glomerular filtration. And of course, glomerular filtration can also be affected by the numbers of nephrons. So if you have less numbers of nephrons, so again, we're talking tons and tons of nephrons in the body, you're also going to decrease your glomerular filtration because you don't have as many to do the blood filtered. So if there's less blood being filtered by the glomerulus, that means that waste products can actually build up. And these waste products that we look at mostly, especially the biggest one, is creatinine, and these are nitrogenous waste products, but also urea. Now, creatinine's never reabsorbed by the kidneys, but urea can be. And so urea is technically also a waste product. So urea, also phosphorus to some extent, and then hydrogen ion. These are the big ones. There's also certain other products that we tend to get rid of a lot. Ammonium is one of them, and then some others that we'll talk about later. So tubular function is actually distinct from the glomerular function, but your tubular flow rate is going to be dictated really by your glomerular flow rate. So remember, your glomerular flow rate is how much filtrate is actually getting across your glomerulus and going into that Bowman's capsule to then become your tubular filtrate. And so if you have decreased glomerular filtration, whether it's due to dehydration or it's due to kidney disease with less functioning nephrons, that would then impact what your tubular flow rate is. And that's when we talk about actual urine-specific gravity and reabsorption of various important substances that your tubules normally reabsorb. So let's talk tubules for a second. So we already talked about the fact that your proximal convoluted tubule reabsorbs a lot of sodium chloride and a lot of water. It also reabsorbs all those other very important things such as bicarb, etc. And so remember that acute kidney injury can really impact your proximal convoluted tubule. It's very metabolically active, so that's one thing to consider. And so you can become dehydrated and lose sodium and chloride there. Remember that you also reabsorb a lot of sodium and chloride in this ascending loop of Henle, which facilitates then your water reabsorption. So that's kind of the second place where you're reabsorbing sodium and chloride. Then the other place was that distal convoluted tubule where you're reabsorbing sodium and chloride but here it's under the effect of aldosterone. So those are kind of the three main places within your kidney where you're reabsorbing sodium and chloride. And only really one of them when we talk about chronic renal failure, although that's not to say that there aren't, or chronic kidney disease, that's not to say that other things aren't going to affect it. But when we talk about the medullary interstitium and medullary nephritis or interstitial nephritis, especially in older cats, and we talk about damage to the tubules, that's this whole medullary interstitial area. That's kind of one concept to kind of consider. And so when you lose that tubular ability to concentrate, and this again can also be an acute kidney injury where you're not able to reabsorb sodium and chloride, or a lack of aldosterone in something such as Addison's disease. But if you have even chronic kidney disease, which damages your tubules and you can't reabsorb, your urine-specific gravity is going to decrease. And so tubules are what determine your urine-specific gravity, for lack of a better way of describing it. And they also determine your solutes that are reabsorbed. And so when we talk about actual inability to concentrate, and we're talking about what's called renal azotemia, that's what we're really talking about, is your tubules. Remember, your glomerulus decides what's filtered, your tubules decide how much to concentrate your actual urine and what it can really reabsorb. Now, the one thing we haven't talked about, again, I know we just did a little bit ago, is ADH. And so ADH, again, works in the distal part of the kidney, but mostly in the collecting ducts. And ADH works to reabsorb water, but can also reabsorb urea. And so half of this saltiness of the medullary interstitium is from that sodium and chloride that's reabsorbed in the ascending loop of Henle, but it's also due to urea reabsorption. And so animals that have a low urea, such as an animal with a portosystemic shunt, can't concentrate as well because their interstitium isn't as salty because they don't have as much urea. So that's just another thing to consider. Again, ADH reabsorbs free water, and it also reabsorbs some urea. And so why that's important is because in a dehydrated animal, where you're stimulating ADH, you can see actually an increased urea relative to the amount of creatinine. Remember, we talked about increases in urea due to GI hemorrhage. Well, it can also be in dehydration, where urea versus creatinine, the urea is greater because ADH is released, and so you're actually reabsorbing more urea. In 2 lectures' time, when we actually talk about acute kidney injury and chronic renal disease or chronic kidney disease, we're going to talk about kind of the differences between glomerular filtration and tubules and how we look at those together. But for now, we're really just going to focus on kind of kidney function and what it's supposed to do. We move on to low sodium and low chloride. We're going to talk about the different places in the kidney and how you can lose sodium and chloride with kidney disease, and then as well as with other diseases.